Good morning, everyone, and welcome to this session. I'm very excited to chair. I welcome, first of all, Mats Hansson to introduce our keynote speaker. We have the honor to guest Maria von Korff. Please, Mats.
Yes, I will introduce the first speaker, the keynote speaker here, and this will be a Ulla Lundqvist Memorial sponsored talk. Ulla Lundqvist was a plant breeder and a dear member of this community, and she passed away in COVID pandemic, the 26th of December 2020. So many of her friends, they couldn't attend the funeral, of course, so instead a lot of money were collected. So they have been deposited at the Royal Academy of Physiographic Society in Lund, one of the sponsors here on the backside. And it was decided that this will go to sponsor a speaker on this conference, and it will be Maria von Korff. A few words about Ulla. She lived most of her life in Sweden, but she was born in Germany, in Göttingen, 1928, but her heart belonged to Austria. And this village, Prince, was very important for her and the family, and they have a very nice house there. And also the mountains here with the Blasen, which is, according to Ulla, the mountain with the richest flora in the Alps. She came from a truly astonishingly intellectual family. So this is her grandmother, Maria Köhne von Marilein. She was studying art, supervised by Gustav Klimt. So if you don't know who he is, he's one of the big painters, big artists. You should Google him. You can see some of his paintings here. The Kiss and many others from the Golden Age. So this is the reason why there is a painting of Ulla's grandmother in Ulla's bedroom in Trins. So now this painting has been deposited to the museum in Vienna. Her grandfather, Anton Köhne von Marilein, he was a professor in botany, and today he would be called a plant ecologist. So he was professor in Innsbruck first and then Vienna, and also curator of the botanical garden in Vienna. And he was growing plants in the botanical garden in Vienna, and then, so that's 160 meters above sea level, in the Trins, in the village, which is 1,200 meters above sea level, and then Blasen, up here, 2,200 meters. So here you have Edelweiss, for example, flowering. And down here, he was growing plants, and now there is a stone there in memory of this ecological field station. And he was communicated his research and discussed that with Charles Darwin. So in the family possessions, there are letters between Charles Darwin and this ancestor of Ulla. The grandfather, he was also a professor in botany, but he was a systematic botanist. Prague and Vienna had very much influence of the academic politics in Hungarian, Austrian country. So he ended up on the 50 shilling banknote in the 60s and the 70s. Ulla's father, Fritz von Wetzstein, he was also a professor in botany, and he was a geneticist. First in Göttingen, then Munich, and then he became director of the Kaiser Wilhelm Institute in Biology in Berlin-Dahlem, which is now the Max Planck Institute for Molecular Biology. So he took the initiative to a Kaiser Wilhelm Institute for Applied Plant Genetics, which was first located outside Vienna. And then during the war, it was moved up to Gattersleben. So that is what you today know as IPK Gattersleben, where we have many people working, present in this conference. But as you see, he died quite early, 1945, from a badly treated pneumonia. And that was, of course, a big loss for the family. So the romantic sound of music life in the Alps there for Ulla and her two brothers, Dieter and Otto, ended abruptly. So they became poor. Ulla was sent to Sweden to a friend of her father, Åke Gustafsson, who was professor in genetics. So Uda called him her second father. And in the other photo here, you can see a group meeting with Professor Gustafsson and his young students. They look quite old, but these guys, they are like 25, 30. So many, at least 4 of these guys here, they ended up as very famous big professors. And for those of you who know Dieter von Wettstein, that's him here. And I think most of you have already realized the gender balance, right? So this photo encouraged me and others then to suggest that this money in this we have here should support a female speaker on this conference. But nobody puts babe in the corner, especially not someone like Uda. She had a very strong character, and she became the queen of Bali mutants. She traveled the world around helping people to phenotype mutants for mutant collections, tilling populations, etc., etc. In the end of her life, at the age of 64, she summarized her research in a thesis. So that was published in 1992, and it's well cited. And she got a lot of recognitions. So she got medals. For example, here she's holding a medal from the Royal Academy of Agricultural Sciences in Sweden. She also got a medal from the International Atomic Energy Association, which is a United Nations thing based in Vienna, and also Accademia dei Giurifili in Italy, in Florence. And this meeting was also extremely important for Uda. So she was the only one that had attended all of them from the very start, 1963. She had a travel grant to come to Latvia if it would have happened 2020. It didn't, as you know. So I know that she would have been extremely proud if there would be a talk by a talented female speaker also working on her mutants. So now it's time for Maria to come up here. And Maria, she's professor in Institute of Plant Genetics and Heinrich Heine University in Düsseldorf. Has also been at the Max Planck Institute in Cologne. And yeah, she's working with Uda mutants. So Uda would have been very happy.
Yeah, thanks a lot, Mats, and also Laura for inviting me. And I feel really very honored to be kind of talking in the memory of Uda, which I didn't know. So it was a surprise to me, yes. I have to say, as Matt said, I probably really owe a lot to her because part of my career was really built on the mutants that she had discovered and described. And I always very much enjoyed talking to Uda because she knew so much about the mutants, but also because this is something that some of you could not have noticed. She spoke this really beautiful and eloquent German that you nowadays don't hear anymore. So I always like to listen to her German. Anyway, so thanks a lot for inviting me. Yeah, so I want to started talking about development, but we already had a great talk by Agatha about development, so you already know a bit about what we are doing. And today I would like to talk about inflorescence development under stress, and more specifically under high ambient temperature stress. Now, and this is, I have to say, still work very much in progress. So we all know that, well, global warming really puts our crops under threat, and it's reproductive development that is particularly sensitive or susceptible to high ambient temperatures. And because it really impairs spike development, floral fertility, and this results typically in a strong reduction of the number of seeds per spike, as I'm trying here to show. Not sure, do I have a pointer here? Is that? Yes. So this is here, you see, this is a spike that develops under optimal condition, and this is the typical form of a spike that develops under high ambient temperature conditions. Now, over the last 2 days, we have heard that plant genetic resources and gene banks are really a treasure to improve stress resilience in our cereals. And one such gene bank, and we've heard about this already, is the gene bank of Icada. And you see here actually a picture I took many years ago. This is the Icada nursery taken from Tel Hadia. So this was the main quarters of Icada, close to Aleppo, which unfortunately doesn't exist anymore. But when I was working there, as I said many years ago, under the Generation Challenge program, we put together yet another barley reference collection. Niels is smiling. So based on passport data and genetic information. So these were 300 accessions that then I took back with me to Germany, single seed propagated it. And today I would like to talk about a subset. So these are only 100 genotypes, mainly land races, and mainly selected to come from origins that are particularly hot and dry. And this collection was genotype with a 50 SNP array. And I have to say we supplemented this or complemented this collection with European barley cultivars. Now we phenotype this collection under controlled conditions. And this is so we grew them under short days. Then we had a period of vernalization because most of the landrace are winter barleys. And then we either grew them under controlled conditions, so this is 20 degrees during the day and 16 degrees during the night, or under high ambient temperature conditions, so 20 degrees during the day and 24 degrees during the night. So we elevated the day and the night temperatures. And we scored a number of many traits, vegetative and reproductive traits. And I just want to present one of them today, or focus, which is major interest in our lab. So this is days to heading as a proxy to flowering time. You see here on the x-axis the different lines, and on the y-axis days to heading. And in blue is a control, and in yellow is the high ambient temperature flowering time. So there was huge variation in days to heading. But if you really looked a bit more closely, there was a certain pattern. We could find that those land races from hot environments tended to accelerate flowering under high ambient temperature conditions, while the elite barley cultivars from Europe, they strongly delayed flowering time, and some of them never flowered at all. They didn't manage to flower. So now, instead of presenting all the other data, I give you a snapshot. And what I'm showing here is the correlation of different traits to seed number. So here you have on the y-axis the correlation coefficients. Here you have a number of traits. So these are the correlation coefficients of these traits to seed number. And what was striking to me is that under control conditions in blue, there is a positive correlation between these vegetative traits, such as leaf size, biomass, node number, height, to seed number. But this was the opposite under high ambient temperature conditions. So here we had a negative correlation. So the more biomass, the less seeds. Now, if we looked at the reproductive traits, we found that the spikelet number, so these are the number of spikelets that you see here are initiated on the inflorescence, were most strongly positively correlated with final grain number under control, but not under high ambient temperature conditions. By contrast, under high ambient temperature conditions, it was really the fertility. So the ability of the spikelet to develop into a floret and then form a seed that was more strongly correlated to final seed number. So this suggested that, let's say, the component traits of yield, cannot really call it yield because it was a controlled experiment, but of seed number, really differed between the two treatments and differed dramatically, I'd say. Now, we had a small population, and we really just found, I would say, 3 major genes that control flowering time in response to high ambient temperature conditions. And one such gene is the one that we've heard a lot about over the last 2 days, and which always comes up, and is the PPDH1, so the photoperiod response 1, which we know under long day conditions induces the expression of flowering locus T, which then, presumably as a protein, moves to the shoot apex to induce inflorescence development, so floral development. We also found that the upstream nighttime repressor of PPH1 is involved. This is early flowering 3, and this is a gene from the circadian clock. And if you've listened yesterday to Kumsal, we also now know that phytochrome C, which is another upstream regulator of PPH1, is also involved in development in response to high ambient temperatures. Now, interestingly, the third gene we found is the vernalization gene, VERN1, which actually under cold is upregulated. So prolonged cold and vernalization is upregulated to then also induce FD1 and floral development. And we found that under high ambient temperature conditions, this gene is downregulated. So it's the opposite regulation to cold, and this causes a downregulation of FD1 or a delayed upregulation and a delay in flowering time in response to high ambient temperatures. So today, because the gene is so interesting, I would like to focus on PPDH1. Now for this gene, there is a natural variant in the CCT domain of the gene that causes an amino acid exchange, and this mutation causes a delay or reduction in the upregulation of FT1 in the leaf, which is then correlated to a delay in floral development and later flowering. So this we know, and this happens under long day conditions. Now, if we go now in the population, so back to the germplasm set, we looked at what happens at the SNP in the PPDH1 that differentiates the two alleles at PPDH1. So this is the A, which is the mutation, it's linked to the mutation. And here you can see that under control conditions, all lines that carried the A flowered later than all lines that carried the G. So this is known. Now, if we look into what happens under high ambient temperature conditions, these late lines were further delayed in development under high ambient temperature conditions, whereas these lines that were fast, so had the wild-type PPDH1 allele, they accelerated flowering. So this is what I believe as a breeder you call a crossover interaction effect. So you had really an interaction of PPDH1 with temperature to control flowering time in this germplasm set. Now that was puzzling, so we wanted to understand a bit better what is happening. And as usual, we took a shortcut. So instead of going into the entire population, what we did is we used and generated integration lines. And this is such an integration line in the background of Golden Promise that you probably all know because we use it for transformation. It's a genotype that is very slow, and it's very slow because it carries the mutation in PPDH1 that makes it late flowering. So we integrate the wild-type PPDH1 allele from the winter barley IGRI, and this is this line that we used. And we also used additional integration lines, so the set that was generated by Klaus in the background of Scarlet that has also the mutation, and then the famous background, the Bowman, and we use the Bowman integration line. Now, all phenotypic data that I'm showing, we could confirm, so I will show for Golden Promise, but we could confirm that in the additional integration lines. Now, when you look now at these two genotypes under control or under high ambient temperature conditions, it is very clear that growth is impaired, so they are smaller, and that's the same in Golden Promise as much as in what we call Golden Promise fast. So this is the line that carries the wild-type PPH1 allele. But what is really, or what was different, was days to heading. So this is Golden Promise, it's very late, and then we could see it's further delayed under high ambient temperature conditions, whereas this Golden Promise fast is accelerated, so flowering gets accelerated under high ambient temperature conditions. So this integration line kind of recapitulated what we found in the germplasm set, so this crossover interaction effect. So now we had the flowering time, but we didn't really know what stage and how is actually the development of the shoot apex changed under high ambient temperature. So what we then typically do, we use the Waddington scale to look at the development of the main shoot apex. Now the Waddington scale looks at the development of the carpel of the most advanced floret on the inflorescence, and that is typically located somewhere in the middle of the spike. So here we can then identify, for example, as stage 2, the initiation of spikelet primordia. So this is the beginning of reproductive development. And as stage 3.5, this is when floral development starts and when the shoot apex starts growing upwards as the stem elongates. What's also interesting is that, as I said, so here we see that spikelet initiation initiates and at some point stops. So this is around stage 4 to 5, depending on the genotype. But then not all spikelets really develop a floret, and not all florets really set seeds. So this is what I simply summarize under floret abortion, but Gustavus Slave already said correctly, this is a bit oversimplified. So there's a lack of development, and then there's also floret abortion. So that in the end, we have less seeds than spikelets initiated. And this depends on the genotype and it depends on the condition. So what really determines the number of seeds per spike is the activity of the shoot of the inflorescence meristem here at the tip that initiates these spikelet primordia on its flanks. And the longer it remains active, the more spikelets are really initiated. And I think as Shander, who just joined my group from Torsten's group, always said, so the number of spikelet primordia that are initiated really mark the maximum yield potential, because it really marks how many seeds can be formed. But then not all of these spikelets really form a floret, and not all of these florets set a grain. So these processes, one after the other, really determine how many seeds you have in the end on the spike. Now, when we then dissected the intergression lines under high ambient temperature and control temperatures, this is what we saw. So you see here on the y-axis, the Waddington stage. I marked here with a line spikelet initiation, and with the other line, this is the beginning of floral development. Now, in Golden Promise, there was a strong delay of floral development. So the major difference was at the later stages. So it was really the floral development that was changed, and there was a delay. The opposite was true in Golden Promise Fast. So it also started to change at the beginning of floral development when there was a strong acceleration. So here we could then see, well, high ambient temperature, at least as it's manifested, has a major effect on floral development. We could also then go and count or score the rate of spikelet meristem initiation. So this is how fast does the inflorescence meristem initiate spikelet primordia, and this is here plotted against days after emergence. In Golden Promise, you see under control conditions, there is about 1.7 spikelet primordia initiated per day, whereas under high ambient temperature conditions, this is very much reduced, almost to half. But then if we go to Golden Promise Fast, we have almost the same induction rate for under control conditions, but this is accelerated under high ambient temperature conditions, which correlates to the much faster floral development. Now, as I said, what is key to the initiation of spikelet primordia is really the activity of the inflorescence meristem that you can see here. And we used confocal laser scanning microscopy to look at morphological changes of the EM over development and in the two conditions. So typically under control conditions, the size declines. And this goes together with the decline or stop of the initiation of spikelet primordia during development. Now, in Golden Promise, we found already strong size reduction under high ambient temperature in both stages, which correlated with the reduced initiation of spike-leaf primordium. So the size difference correlated with a reduced initiation of spike-leaf primordia, whereas in Golden Promise Fast, we did not see such difference between control and high ambient temperature conditions. So we have a difference in the initiation of spike-leaf primordia and thus in the maximum yield potential of each spike, so the existence of spike-leaf primordia. But we could also see that there's a strong drop, as I said on my first slide, in the seed set. And this is what you see here. So this Golden Promise, under high ambient temperature, there are only sometimes 3 or 4 seeds. It looks really sad. Whereas seed set was much better in Golden Promise Fast under high ambient temperature conditions. And this is just shown here. So this is the fertility score. This is the number of grains that are formed per spikelet or per floret. So it's a ratio. And we have a strong drop here in Golden Promise as compared to the introgression line. We wonder then whether it's the male or the female organs that are most susceptible to ambient temperature. And for this, we did cross-pollination experiments. So we either grew the ovary at 28 degrees and used pollen from 20 degrees, or vice versa. And you see here such a spike. So this is an ovary grown at 20 degrees, self-pollinated on the side here with pollen also from 20 degrees. And then we emasculated on the other side and pollinated with pollen from 28 degrees. And you can see very low seed set. So we quantify this, actually for two here, I have 2 pairs of introgression lines. That is also scarlet here. And you see here in blue is the drop of fertility if the if the carpel was grown under 28 degrees. So there was a susceptibility also of the female organ. And in light blue, it is the drop in fertility if the pollen was grown under 28 degrees. It was even much stronger. So these are the 2 spring barleys here. And these are the introgression lines. And you see that the drop in fertility is much reduced, which we think is due to the introgression of the PPDH1 allele. Now, when we looked at the pollen, we could see that under high ambient temperature conditions, a lot of the pollen was completely shrinked and empty, and likely therefore not fertile, in Golden Promise, whereas in Golden Promise Fast, most of the pollen looked fine, so no big difference between control and stress conditions. So with these phenotypes, of course, we're wondering what are the molecular changes in the meristem downstream of PPDH1 that could explain these phenomena. For this purpose, we did RNA-seq profiling using these 2 genotypes, Golden Promise Fast, at actually 4 stages. So the vegetative stage, at the onset of spikelet primordia induction, at the beginning of floral development, and during floral development, so around the time of meiosis. And we sampled shoot apices and also leaves. And today, we haven't really analyzed this last stage yet, so I will focus on the first 3 stages. We used actually the BART reference for mapping the reads, and that allowed us to map more than 90% of the reads, so that was really a great resource. I think Ronja Wonneberger just introduced that new resource. And we scored differentially regulated genes for every stage and genotype separately. Now, if you look at this, what was striking us, of course, at first sight, is that there were many more regulated genes in Golden Promise than Golden Promise Fast. It's really a huge difference. So then we had about 3,000 genes that were regulated by temperature. But then we argued we are more interested in those genes that are also regulated by development, because these are we can presumably link to the differences in development under high ambient temperature conditions. And these are about 400 genes. Unfortunately, only a bit more than half of them are really annotated so far. So we are really hoping, and we're also trying to improve that. So when we clustered the samples, we could see there was clustering exactly according to the stages. Not the stress treatment, but really the stages. So we have here stage 1, stage 3.5, and 2, with the exception of the golden promise samples under high ambient temperature conditions. So they really clustered very differently. And we used gene clustering, and you see here 2 clusters, and these are genes that are particularly downregulated under high ambient temperature in golden promise. And these are the cluster of genes that are specifically upregulated under high ambient temperature conditions in golden promise. Now, the analysis of the data is still very much ongoing, but I just want to give you a snapshot of what we found. So luckily, we have some information on I have to say, only some information on the regulation of development in barley. Now, we know that PBD21 induces the expression of FT1. So, and this is a gene that is only expressed in the leaf, so it's all happening in the leaf. So when we looked at the expression of FT1 in the leaf, we found the following. So here, maybe to explain, this is transcript per million. You have here the 3 stages. And then in yellow, this is golden promise. And in blue, this is golden promise fast. And then the dashed line are the samples from the high ambient temperature treatments. Now we could recapitulate what I already told you. There's a big difference in FT1 expression between these 2 genotypes, which is then linked to the difference in development. But what we could find is that in both genotypes, FT1 is downregulated, but still the expression in golden promise fast, even under high ambient temperature condition, is much higher than in golden promise. Now, there's not only one FT gene, there are many FT-like genes, and one interesting one is FT2, which is expressed in the meristem. And here we have a very similar pattern. So we see a downregulation under high ambient temperature condition, which at least in golden promise correlated with a delay in development. Now we know from work in Arabidopsis and also in tomato that what really matters for the development of the inflorescence is not the expression of individual genes, but it's really the dosage of genes. So the dosage of inducing versus repressing genes, and such inducing genes are the flowering locus T-like genes, such as FT1 and FT2. And such a repressor is centroradialis that Eyal yesterday already introduced. So it's really, so if you have more FT against Sen, the spike tends to differentiate. The meristem tends to stop working. Whereas if you have more Sen, then you keep the meristem alive. So this is the balance that you need. So what we then did, we looked at the balance or the ratio FT1 to Sen, and here we have to use the FT1 in the leaf, which we think is transported as a protein into the shoot apex. And again, what you can see here is a big difference between the genotypes, a downregulation under high ambient temperature, but here actually the ratio became higher versus FT in the golden promise fast, which correlates to the faster development of this genotype under high ambient temperature conditions. And we could also see this in for FT2 that is expressed in the meristem. Now we are now waiting for the samples of stage 6 to see whether we can confirm this ratio change that perfectly correlates with the observed differences or this crossover interaction effect that we see in terms of development. Now, I also told you what we found altered was the size of the inflorescence meristem. So we looked at those genes that we know that are involved in the maintenance of the inflorescence meristem. One of them is the INT-MAP2 that we have shown last year to be really crucial to maintain the EM. And we see it's downregulated typically, but it was more strongly downregulated under high ambient temperature conditions. So high ambient temperatures, they cause a downregulation of AP2, which then correlates with a faster stop of the activity of the inflorescence meristem. We also identified a scarecrow transcription factor, which was particularly low in golden promise under high ambient temperature conditions. This gene is very important to maintain the stem cell niche, so to maintain the meristemness of the EM. And we identified the trehalose 6-phosphate synthase that is important to synthesize trehalose 6-phosphate and this is a sucrose signaling metabolite that is important to signal to the plant the sugar status and really induces floral development. That also was strongly downregulated, in particular in golden promise under high ambient temperature conditions. Now, our last set of genes that we thought was interesting are those that really initiate the differentiation of floral organs at the side of the inflorescence meristem, and these are the Matchbox genes, and they were also strongly downregulated, in particular in the golden promise under high ambient temperature conditions. So this related to the very much slowed down development of the floral organs. Just the last set of genes where we identified lots of hormone-related genes. And here I just show the auxin-related genes because we know they're important for the initiation of new organs, so floral organs. And also these were most strongly regulated in golden promise under high ambient temperature conditions. Now we want to confirm that hormone homeostasis is altered by high ambient temperature conditions, and we've started to do so using fluorescent reporter lines. This is such a fluorescent reporter for PIN1, which is important for polar auxin transport. And you can see here its expression in golden promise under control and under high ambient temperature conditions. So it's always this exactly same conditions that we are looking at. And you see it's strongly downregulated. Whereas this is not the case in golden promise fast, and here they see the quantification of the signal. So this suggests that really auxin transport, auxin maxima might be changed. And here we teamed up actually with Laura Rossini and Raffaella Battaglia, and we're generating additional fluorescence reporter lines, so for example DR5, and also for cytokinin. And we've crossed them with the two genotypes to understand to what extent PBDH1 and downstream genes alter the homeostasis of auxin and cytokinin. So with this, I would like to summarize. So we found large variation in this nice germplasm set for flowering time or response to high ambient temperature. And what I didn't show, it also kind of extended to the ability to set seeds under high ambient temperatures. What we see is that, and we know that from other talks, PBDH1, or there was strong pleiotropy, so high temperature and PBDH1 really affected the development of different meristems. So inflorescence meristem, but also the spikelet and the floral meristems. And that was linked to downregulation of meristem maintenance and differentiation genes. We think that the hormone homeostasis is altered, but here the confirmation is pending. And as we see that so many different meristems are altered in a certain correlated way, we started to think instead of looking at whole inflorescences, we need to dissect the tissue, or what happens in a specific tissue, in a specific meristem, so EM versus, so the inflorescence versus the spikelet versus the floral meristem, to identify downstream, more specific networks and to disentangle this. And this is something that has been started by Edgar de Meso Arevalo, and he's started to do single-cell sequencing in the developing inflorescence meristems to identify specific cell clusters. And he's developed a method together with a company, molecular cartography, that is a substitute to this very laborious in situ hybridization. We can actually use probes to localize the expression of specific genes. This is, for example, a Matchbox genes, to specific parts of the inflorescence, which we hope will allow us to dissect much more the molecular networks that control the individual meristems that we have on the inflorescence. Now, with this, I would like to close, and I would like to say most of the data that I presented today was generated by Tianyu Lan, who is in the audience. So this is her here. You will recognize her, hopefully. She has poster number 18. So if you're interested in the work, please go and see the poster and talk to her. There's also Gesa, who's contributed. She's working on the FT1. She has poster number 63. And of course, Edgar, that I just mentioned the work. So if you're interested in that, please go and see poster number 67. Now, before I close, I would like to acknowledge Michael Baum and Salvatore Ceccarelli, whom I worked with when I was at ICADE and who allowed me to take this collection back to Germany, and who were always very inspiring and actually brought me back to Bali because I had a small detour into Arabidopsis, but both of them really brought me back to Bali. And I would like to acknowledge our cooperation with Laura and Rafaela and Veronica on the fluorescent reporter lines, and also Rüdiger Simon, who's a colleague of mine at the HHU. And I thank you for your attention.
Hi, Mary. Thanks for a nice talk. My question is about the FT. So since FT has been reported to travel through the vasculature, and do you believe in such earlier stage or the spikes that the vascular system is already been established, or maybe it's a different mechanisms?
I'm not sure if I fully understood your question. Is it whether FT is involved in the development of the vasculature?
No, FT is traveling through the vascular system. And,
Okay.
For example, before Weddington 3.5, is this system already been established in the spikes?
Yeah, so I mean, I was saying presumably because it has been demonstrated in Arabidopsis and rice, and that has never really been demonstrated, the traveling of the protein in Bali, so I'm being cautious. But we believe that this is happening, and it is happening actually, so it must be happening before the spikelet initiation, because this is something that doesn't necessarily depend on this FT1, but as I said, we have several FT-like genes, and you need to have at least one of them expressed to get the spikelet initiated. So, yeah. So I cannot say for sure that the protein is traveling, but it's been shown in rice, so we believe that this is also true in barley.
Hi, Maria.
I'm not seeing you. There in the back. Okay.
Thanks for the nice talk. In wheat, it has been shown that the PPD1 mediated FT1 delay in the expression leads to supernumerary spikelet formation. Do you know if that is also true in barley? Did you check for this phenotype?
Yeah, so maybe you have to talk to Kumsal later. So we also have some floral aberrations. So we sometimes see more different ways of producing more seeds per spikelets. But then how this happens is sometimes a bit different. So sometimes it's a double florets. Sometimes, like in Kumsal's case with a 5C, it's really that two carpels are formed. So these are floral abnormalities, I would say. But they really form two seeds, and both seeds germinate. Yeah, so we do see this depending on the genotype and condition. But in this case, actually, it's linked to faster development, isn't it, Kumsal?
Yes.
Yes.
The faster.
So I think the work by Scott Bowden showed that it's really when you delay development, you tend to get this double seed, whereas we tend to see it more when we accelerate. So it seems to be a bit different. I cannot tell you why, but this is what we noticed. Yeah.
So one last question, Niels.
Yeah, I actually have two questions. So you applied the high temperatures from very beginning on, yeah? So after taking them from vernization, the initial experiment, but also after you, with the introgression lines, you work really all the time with the high temperatures. Have you done experiments also to apply this later, and to which extent does it overall change the pattern, or is it just delaying the signal? And the second question, you didn't like to talk about VRN, but your initial observation was in winter barley. And the question is whether this high ambient temperature directly after vernization is just reverting the vernization effect.
Yeah, so I start with the last question. So that's true. We see an allele-specific effect. So this reversion is much stronger, obviously, with the winter type allele than with the spring. Yes, so it really, so we see an upregulation, but then it's downregulated again, depending on the genetic background also. So this is maybe something that, people used to call devernalization. So if you move plants out of the vernalization, maybe the vernalization wasn't complete, and then you move them to high ambient temperature conditions. This is a phenomenon that's been described as devernalization and is probably linked to the downregulation of EN1. So it seems to react to both to cold and to high ambient temperature conditions in an allele-specific manner. Yeah. The first question, yes. Also, maybe you have to talk to Tianyu later. So we've done lots of experiments by shifting plants at different stages to high ambient temperature conditions to understand which stage is most susceptible. Actually, I said this is a floral development that has most changed, but Tianyu has done experiments where really crazy. So she was putting them at a very early stage, so the vegetative stage under high ambient temperature conditions, and then back to normal conditions, and you still see the phenotype. So it seems that there's also a memory, so it's not lost. So even if you treat the plants very early, you see the same effect on floral development much, much later. So this delay in development. But we've also exposed them at later stages, and I have to say, the effect becomes more drastic. If you grow the plants for a longer time under control conditions, and then around floral development, you shift them to high ambient temperature conditions, the effect is the same, but it's stronger. Yeah. But we didn't see big differences. So first we thought it's really only floral development that is susceptible, but I think, as we've done it also very early, that, I mean, barley doesn't forget. It's like Sonia knows, if you overwater, so if there's waterlogging in the first 2 weeks, you can see it 8 weeks later. So this is something that, yeah. So I always say barley doesn't forget if you don't treat it well.
Yeah, thank you, Lara, for the introduction and also selecting my talk for the presentation, selecting my topic for the presentation. Yeah, today I'd like to present briefly about some of the results that I have been getting from the studies from one of the barley spike developmental mutants. The mutant, it's called multiflorus 2, and yeah, this is a very interesting mutant because the spikelet determinacy in this mutant is altered so that in place of one floret per spikelet, you start to see multiple florets, and also occasionally there is a grain set on the secondary florets. And throughout the presentation, I will touch upon one of the most interesting and underexplored inflorescence organ, that is rachilla, and how the determinacy or indeterminacy of this important organ can dictate the floret number per spikelet in general in the Tritiaceae species, and also in the grasses where the spikelets are harboring multiple florets. So what is determinacy, inflorescence determinacy or indeterminacy? So this can be defined based on the images that you are seeing on the screen. So the one on the left side is a determinate inflorescence. So here you have a definitive number of flowers on the determinate inflorescence, and this is mainly because of the terminal flower that is forming on the inflorescence, and this doesn't let any other flowers to form, and that's why you see definitive number of flowers on the determinate inflorescence. And in case of indeterminate inflorescences, there is no terminal flower formation, and the inflorescence can produce multiple number of flowers, but during the course of the development, this is no more possible, mainly because of the assimilate problem. So, of course, the inflorescence meristem itself is determinate, so it can form multiple florets. So these are the two inflorescence types that you generally see in the nature, but if you can imagine, if you can remember looking at the inflorescences, there is a tremendous variation, and these are not just the two types of inflorescences that you see in the nature, and the majority of them are derivatives of these two inflorescence types. The one on the left side is the wheat inflorescence. It is a determinate inflorescence, mainly because of the formation of terminal spikelet. But on the other hand, if you look at the wheat spikelet itself, it is indeterminate structure. So because it starts to produce multiple florets, at least you can see 6 florets in the wheat spikelet. And in contrast, the barley inflorescence is indeterminate because the apical meristem, the inflorescence apical meristem is indeterminate, and it can continuously produce more number of spikelets towards the apex. And on the other hand, the spikelet itself is a determinate structure, and it can produce just one floret per spikelet. So this crucial difference is coming due to the organ that I have been telling, that is racula, which is highlighted in blue color here. If you look at the wheat spikelet racula, so it is a kind of indeterminate structure, and it starts to elongate after producing first floret. And at each articulation junction of the wheat floret, you start to see a floret formation. And however, this is not true in case of barley. So what happens is the floret, after first floret formation, the racula ceases to grow, and there is no more elongation, and it completely stops growth. That's why you see a single floret in barley. If you look at the actual spikelets of barley and wheat, so this is how they look like. So in both 2-rod and 6-rod barleys, the racula elongates to produce just one floret. So the racula is highlighted in yellow color here, the small hairy structure that you see here. And whereas in case of wheat, so you can see multiple florets, as I told. So here is the racula that is highlighted in yellow color, and at each articulation junction, a floret is formed. Actually, the florets are stripped off in this spikelet, so that's why you don't see any florets. But wherever you see a blue color, that indicates a floret on the spikelet. So, in principle, even in barley, there is a potential to form more number of florets per spikelet, but the genetic mechanism within the barley suppresses this to just form one floret. So, however, there are, over the course of time, I found various mutants in barley which can revert a determinate racula to indeterminate racula to produce more florets per spikelet. So, a few of them are the branching mutants that are compositum 1, compositum 2, and 6-rod spike 4, and low number of tellus 1. So, there is a peculiar feature in all of these mutants that, so you all know that barley has 3 spikelets per node. So what happens in case of central spikelets and lateral spikelets is quite different in these mutants. So in case of central spikelets, so the racula transforms into a kind of branch-like structure. That means the racula is reverted into a kind of spike-type inflorescence. And only the lateral spikelets, the racula elongates to produce a secondary floret. So here you see the ones that are highlighted in blue arrows, they are secondary spikelets coming from the lateral spikelets. So this is a common feature in all of these mutants. So what I would like to say is there is a potential of the barley racula to become indeterminate, but somehow the genetic mechanism suppresses this feature in barley. And this is also true in wheat. So there are a few studies that have been reported in wheat where the indeterminate nature of the wheat racula can be further enhanced by modulating the genetic mechanism underlying this. So one of these main genes that have been identified that can regulate the racula elongation in wheat is the Q gene, and the associated miRNA-172, which can regulate the Q gene. So when there is a misregulation of either of these, there is a chance to produce more number of florets within spikelet. So also recently in Maria's group, they have identified this epitala2, which is ortholog of Q, also as a kind of important regulator of terminal spikelet, a kind of negative regulator of terminal spikelet formation in barley. So also, I think in this mutant also there is a regulation for racula elongation process. So I think this has to be further explored. And coming to the multiflorus 2, the mutant that I have been working. So here, these are the phenotypes for this mutant. So what we see on the top is the wild type, where you see single floret per spikelet. But in case of mutant, what is happening is the lateral spikelet region, the racula elongates further to produce a second floret. So, and in some cases, it also generates a third floret, like what you see in the right side bottom picture. and this is how it looks like along the spike. So there is a kind of uniform development of additional spikelets that are coming from the racular elongation in lateral spikelets. So, and frequently they are setting grain. So what one can say is the lateral spikelet racular elongates to produce up to 3 florets in this mutant. And one more interesting phenotype that I have observed in this mutant is the formation of supernumerary spikelets. So here the supernumerary spikelets are highlighted in green color. So the ones in the blue are the racular elongation that leads to extra floret formation in the lateral spikelets. So it's a little bit complicated phenotype. So at the end, I can summarize the phenotype of MUL2 is a kind of lateral spikelet indeterminacy that leads to secondary floret formation in the lateral spikelets and also supernumerary spikelet phenotype in the central spikelets. So it's a combination of these two phenotypes. Yeah, we wanted to exactly study when these developmental abnormalities are happening in the mutant. So for this reason, we followed the developmental pattern. So, so the one on the left side, the micrograph on the left side is from wild type, and on the right is the mutant. So the racula in this wild type is racula of lateral spikelets in this wild type is highlighted in yellow color, and that of the mutant is in blue color. So if you can closely look at, there is already a fasciation of racula meristem in the lateral spikelets, indicating that there is a chance that this racula meristem can potentially elongate to produce a second floret. And this becomes much more clear in the later stages. Like if you see on the extreme right side, the racula in the wild type, which is in yellow color, is almost suppressed in the growth. But whereas in the mutant, the racula further elongates to form a second floret in the lateral spikelets that are indicated in the blue color. And the actual racula itself, you can see on the adaxial side of the secondary spikelet that is indicated in the blue color. So the developmental destiny for the racula is decided quite early in the development of barley spike development. So as I told, the barley, the MUL2 mutant, it also shows formation of supernumerary spikelets. So if you remember from one of my colleagues' presentation yesterday, Go Jing, so he also showed a mutant that is called FLO-A, and there is a phenotypic similarity between FLO and MUL2 because they both form supernumerary spikelets in the central spikelet region. So we were wondering whether these two mutants are allelic to each other. So what we did, we made a cross between these two, and the F1s, actually they are saying that they're allelic to each other because you see the extra spikelet phenotype in the F1s. However, we wanted to be more sure whether this is true, that these two are allelic to each other, and then we screened the F2 generation, and so the results from the F2 are completely refuting the data from the F1. So in the F2 generation, there is a kind of segregation for this phenotype. You see completely wild-type phenotype and also different levels of supernumerary spikelet phenotypes. So this clearly says that the allelism data that you are seeing in the F1 is cannot be believed, and in fact, the phenotype, the supernumerary spikelet phenotype that you are seeing in MUL2 could be potentially coming from a different gene. So this data says that there may be potentially more genes that are regulating the multiflorus 2 phenotype. That is, you have the lateral spikelet phenotype, that is racula elongation and secondary spikelet formation, and this could be regulated by one gene, and the supernumerary spikelet formation could be regulated by a secondary gene. So for mapping this phenotype, we developed F2 mapping populations by crossing MUL2 with Morex and Moncalm. And before going for phenotyping, what I did was I did a kind of detailed phenotypic analysis of this mutant so that it would be easy for screening the F2 populations. So upon careful phenotyping of this mutant, we observed a kind of range of phenotypes of lateral spikelet racula elongation. So in some cases, there is a complete grain set in the secondary florets. In some cases, there is just a floret with on elongation. And in other cases, there is a secondary floret without on elongation, and in the other cases, there is racular elongation, but there is no sign of floret formation. So this data clearly shows that it could be a kind of quantitative phenotype that one can expect in the mapping population. And on top of that, the other mapping parent that we used, that is Morex, is also showing a kind of weak racular elongation phenotype, that is the class 4 phenotype, where you see hairiness, hair-like racula or wedge-shaped racula. So this clearly indicates that in this population, the racular elongation phenotype can be quantified as a kind of quantitative trait. So what we did, we have quantified the numbers of different racular elongation phenotypes, and we treated this as a kind of QTL, and we mapped this as a quantitative trait. Yeah, these are the frequency distributions of the different phenotypes of lateral spikelet indeterminacy and central spikelet, supernumerary spikelet phenotypes. I mean, the histograms, they don't show a kind of normal distribution, so you may not expect a kind of a big, several QTLs, but still it is possible that the phenotypes can be regulated by a few number of genes, maybe 2, 3, like this. So, and then we proceeded with the QTL analysis, and the result was very interesting. So for the lateral spikelet racular elongation phenotype, we found at least 2 QTLs on chromosome 2 and chromosome 6. And on top of that, we found a minor QTL on chromosome 3, and in case of 2H and 6H QTLs, the main phenotypic effect is coming from the MUL2 genotype. And in case of 3H minor QTL, the phenotypic effect is coming from Morex. So in the previous slides, I have already indicated that Morex also has a kind of racular elongation phenotype that leads to secondary floret formation. So we have identified 3 QTLs coming from different parents. This is with respect to the lateral spikelet phenotype, and there is also a kind of central spikelet, supernumerary spikelet phenotype in this mutant. So an interesting thing is, when we map this central spikelet, supernumerary spikelet phenotype, there is a single QTL on chromosome 6H, and it exactly collocates with the second QTL of lateral spikelet indeterminacy phenotype. So this clearly indicates that there is a kind of interaction between these 2 QTLs, and potentially the phenotypes are regulated in a dependent manner by these 2 QTLs. To check the interaction effects of these 2 QTLs, we compared the phenotypes of 2H and 6H QTL alleles. And on the left, the phenotype is coming from lateral spikelet racular elongation phenotype. And if you see the one on the left side indicated in blue color, these are in this case, the phenotype is contributed by the mutant alleles at both chromosome 2 and chromosome 6, and there is a clear increase in the phenotypic effect when there is a mutant allele at both QTLs. However, when there is a wild type allele at 6H QTL, there is a strong reduction in the lateral spikelet indeterminacy phenotype. So this clearly indicates that these 2 QTLs are important for contributing to this lateral spikelet racular elongation phenotype. However, that's not true for the central spikelet, supernumerary spikelet phenotype, and this phenotype seems to be exclusively conditioned by the 6H QTL, and there is no influence of 2H alleles at this locus for this phenotype. So at the moment, we are trying to Mendelize these 2 QTLs because it's very highly impossible to clone 2 different QTLs in a single population. So what we are doing is we are identifying lines that are fixed for each QTL and are the lines that are exclusively segregating for one of the QTLs, and we are trying to follow this by map-based cloning. Yeah, this is I have submitted a proposal based on this work, and actually it's rejected. I'm going to resubmit it again. So let's see. So this is with respect to MUL2, and we were also interested in identifying more developmental mutants that are important for racular elongation process. So for this reason, we have screened a large number of induced mutant collection from the Nordic barley genetics stocks from Norgen. So I found two interesting mutants that are having a problem in the racular elongation or racular determinacy. So one of them is, if you see the scanning electron micrographs here, so what you see here is the ones on the indicated with the green star are the central spikelet regions, and the one with yellow stars are the lateral spikelet, actual lateral spikelets. So in the middle where you see this green star, actually it's a central spikelet, but there is no sign of spikelet formation. And what you are actually seeing is the racula that is continuously elongating. So, when you look further, or when you look further into the development, this racula is potentially forming a kind of spike-like structure. So this indicates that the racula has the potential to form a kind of complete spike-like structure. And if you remember the higher order inflorescences like panicle inflorescences of rice or sorghum, so the spikelets are formed on the secondary axis. So in my opinion, the racula is a kind of secondary axis which has the potential to form a completely independent spike. So at the moment, we are generating populations.
Ravi, 1 minute, really.
Okay, sorry. So at the moment, we are developing mapping populations for this. And the second mutant is also a kind of racula elongation mutant, but this is in the MUL2 case, it's the lateral spikelets that are getting indeterminate, but here the central spikelets are getting indeterminate, and we are also developing mapping populations for this, and in order to identify the genes responsible for this phenotype. So to summarize, the barley multiflorus 2 is a combination of lateral spikelet indeterminacy and central spikelet supernumerous spikelet phenotypes. And we have identified 2 independent QTLs and 2H and 6H. The central spikelet supernumerous spikelet phenotype is majorly controlled by 6H QTL. And phenotypic screening of 6-rod barleys reveals. So I haven't touched this upon, but we have also screened a large number of 6-rod genotypes, which out of which 288 of them showed racula elongation. So potentially these are alleles of the MUL2, and this. So we have already the alleles available for the phenotype. And of course, the identification and molecular mechanism of these racula elongation and floret formation mutants can help augmenting the grain yield, Triticea and probably other cereal crops. So with this, I would like to thank several people who are involved in this, and I also would like to thank several people who have developed barley mutant stocks, so which have been very useful for my research over the past 12 years. Thank you.
Okay. Right. I'd like to kick off by actually going back to a slide from Martin Mascher's talk first thing on Monday, and basically showing the nice effect of the large inversion on 7H in planet, and the effect that has on the 2 genetic maps, whether the presence or absence of that inversion. Yeah, can you hear me? Okay. So basically the effect of the inversion on the 2 genetic maps. And the point I just wanted to make is that a genetic map is a phenotype. It's derived from the patterns of recombination events in a population, but it's controlled by genetics, it's affected by environment, it's in this case strongly affected by the genetic background. So the pattern of recombination events themselves result from complex spatial temporal progression of chromosomes during meiosis. And it's an incredibly complex developmental process that occurs in the developing inflorescence at about the Waddington stage 6 within the boot. And you can see here the progression of meiosis from replication through prophase 1 through to metaphase 1 through to the development of gametes at the end of, towards the end of the slide there. And you can see particularly the exchange of information associated with crossovers that within prophase that become evident as chiasmata during metaphase 1. Fortunately, quite a lot of it is known about meiosis and recombination, mostly through model eukaryotic systems. You can study the progression of meiosis in barley cytologically. You have the tools and the expertise. And barley is a well-behaved eukaryote. It goes through prophase, metaphase, prophase 1. The combination occurs. You can see the zipping up of the chromosomes in pachytene through to diploidine. And again, you can see the bivalence at metaphase 1 showing the chiasmata, showing the crossovers. What's interesting about barley and related species is that recombination is fairly limited, certainly in comparison even to Brachypodium. And we were interested whether this is a constraint to breeding progress, to evolution generally, and whether there's means to influence those patterns of recombination and the rate of recombination, and whether that would be of potential application in breeding. Fortunately, in barley, there's a large resource that we can use, and we are building on previous work, and particularly for this work I'm going to be producing. This builds on work of 50 years ago by Tom Ramage in the University of Arizona, who characterized a large number of spontaneous mutants in Betses, which had a semi-sterile phenotype and that had a clear meiotic phenotype. And here you see the effect of one of these mutations on the patterns of crossing over at metaphase 1, where you see in this particular blown up cell, you can see the spread with 2 ring bivalents, 2 rod bivalents, and then a number of univalents. In particular, again, a lot of work was done, and at least 15 independent loci were described. Again, all this is in the Biogenomics newsletter, and 24 of these mutants were then integrated into the Bowman neoisogenic lines. Thanks, Jerry. And we then took some of those, 15 of those Bowman desynaptic neoisogenic lines and produced biparental F2 populations. Just concentrating on one particular desynaptic 10, we then used a traditional map-based cloning method to identify the underlying gene. But again, I'd just like to point out on the metaphase 1 slides on the second box on the right here. In the wild type, you have 7 ring bivalents. In the DES10 mutant, I'm not sure you can see it particularly well, you have a disrupted chiasmata phenotype with the presence of rod bivalents and some univalents as well. We were able to finally identify the gene. It's caused by, the mutation is caused by an exonic deletion in the barley ortholog of MLH3, which is a well-known mismatch repair protein that has a role in the resolution of double homology actions. And the deleted exon knocks out the majority of the conserved metal binding motif that is essential for the endonuclease activity of the gene. So this is, I mean, it's great, we've got it published, but DES10 reduces recombination, as was clear from the meiotic phenotype. Essentially, it halves the amount of recombination that results from process. So this is interesting academically, but doesn't actually give us a handle to help to increase recombination. So what we did was use it as a suppressor screen. It took a while to bulk up the seeds because it's semi-sterile, and then mutagenize that with a range of levels of EMS, which we previously shown to be give a useful degree of mutation. These families were then grown out in a polytunnel and screened for increased fertility relative to the background. And that's easy to write, but in fact, when we were doing it, I was questioning our sanity. It's not an ideal background to screen for mutation. We had some volunteers coming through as well, which didn't help matters. But fortunately, Bowman's got a smooth horn, so we were able to distinguish the volunteers from the DES 10 plants. However, after a lot of work by my colleague Malcolm Macaulay, we did actually find 10 M4 plants with restored fertility. We then used those to screen those with a hybridization capture MyBates pool that we developed for the shuttle program, and these were concentrated on previously described meiotic genes. And fortunately for us, the line which showed the highest level of restored fertility contained a point mutation in the coding sequence of the body homolog of RECQ-like4 helicase 4. So, which is a known it's a DNA repair gene well known with anti-recombination properties. This mutation we found produced a non-synonymous mutation in the conserved helicase domain. So, and it was predicted to be deleterious. So we were pretty certain we had the underlying mutation that was causing this restoration of fertility. So then we crossed it back onto wild type, in this case Barker, produced an F1, and then tracked the alleles at both the MLH3 and RECQ-like4 loci with Cas markers. And we ended up with an F3 population that we were able to, F2 population, which we were then selected for the homozygous classes. So we ended up with 4 classes, which we then used to produce F3 individuals from those F2 families. So we ended up with populations essentially with wild-type alleles at both loci, mutants, ones with only a mutation in RECQ-like4, or only MLH3, or the double mutant. When we should have included a slide with some information here, but when you looked at the fertility of those populations, there was a clear fertility effect in the single MLH3 mutant, but the other three populations were essentially the same. You can, when we looked at the cytologically, you could see the wild type metaphase 1. Metaphase 1 showed again 7-ring bivalence, the wild type, whereas the MLH3 mutant lines with only the MLH3 mutation showed again this disrupted meiotic phenotype with the presence of either rod bivalence and some U bivalence as well. Excuse me. However, for the lines that contain the RECQ-like4 mutation, both seem to have something close to a wild-like phenotype, although there was the suggestion of maybe some additional chiasmata, some additional bumps on these rings that potentially could be additional crossing over events. There you go, sorry. When you did the genetic mapping, however, it was clear that we were able to, we did the genetic mapping of these F3 families with the 50K SNP chip, and this visualization essentially aligns all the 7 chromosomes, puts them together, and elongates them to the same length. So essentially, you're looking at the summation of the body genome as one chromosome. And when you look at the total amounts of recombination across this barley chromosome, you can see the wild type amount of recombination in physical intervals of the genome. You can see it that you have a strong distal distribution of recombination as in green. When you look at the MLH3 population, as expected, you have a much reduced recombination, as was evident from the metaphase 1 spreads. However, when we looked at the two populations that contained the Reculite 4, we found that essentially we had twice as much recombination as you found in the wild type. Interestingly, we couldn't actually see a significant difference between the double mutants and the lines with the Reculite, the single Reculite 4 mutation, which was somewhat unexpected, but but potentially might say something about the mechanisms of the interaction of these two genes. To just quick summarize in that case, we've isolated a novel mutation in Reculite 4 that from a suppressor screen that worked of a semi-sterile MLH3 background. The recombination levels we found were more or less double compared to wild type. This corresponds to some other work in other systems and Arabidopsis work from the group of Raphael Mercier. But it's clear this does give a potential means of manipulating recombination within barley. At present, we're back crossing that into other backgrounds to check the effect, but there's also some interesting meiotic implications of work on the double mutant. I'd like to acknowledge the group, in particular Isabel Koller, who did the cytology, Mikhail, who did a lot of work on the Reculite 4 work, Miriam, who did a lot of the bioinformatics and the design of the pooling strategy, and Malcolm for the work on the screen. Thank you, and I'll take any questions.
Thanks, Luke. If I got it right, the increase in recombination rate was still in the gene-rich distal compartments. Do you have any hope that you might be able to increase it in the rest of the chromosome?
I have some hope, but it's less than it was when I started this work. You can influence patterns of recombination through heat stress, but it's minimal. And almost all the effects we have seen have been tended to be distal. We have other mutations, again, that do positively affect recombination, but again, they tend to be distal. I think it's something fundamental, more to do with potentially the timing of replication and the duration of meiosis, rather than necessarily individual mutations in some of the obvious genes. I think we're going to have to look at it's something very fundamental to the organization of the POIG, and probably relates to the basic karyotype number of 7 and the amount of retrotransposition and everything else. So I think there's some very strong constraints on changing that sort of pattern of recombination in those sort of areas. That would be my guess.
Thank you, Luke. Smart idea to find this mutant. Have you seen some interest of commercial breeders to introduce that into their material, or are you offering that?
We're in discussion. I mean, that's certainly something we'd be interested to explore. I mean, we are unsure. Again, the level of recombination is fairly what's the word? Conserved across most crosses. When you look at most genetic maps, they're generally the same sort of length from distinct, even in wild crosses, wide crosses. So potentially there's some constraint, again, evolutionary, to give you that sort of recombination range. But again, whether what's the effect of introducing more recombination into a breeding program is as yet unknown. Whether it does help on your potential gains or rate of gain, or whether it produces other problems in the maintenance of desired combinations, we really don't know.
Mark?
Hey, look, I don't know anything about these desynaptic mutants, but you found DSP10 to be an enzyme. Can it be mutations be rather in the structure of the DNA, so they behave the same way? Is that a possibility?
Like, could you repeat?
I mean, yeah, could it be not that they are coding something, doing something in recombination? Could it be the structure of the DNA itself doing something so they look like this kind of mutation?
The structure, what we're seeing the on the gross, were you talking about the gross conformation of the chromosomes, or whether?
Yeah, or the chromosomes, how they align and so on, so they can behave in this way.
I mean, certainly inversions do impact on recombination and on the behavior of the chromosomes as you progress through meiosis. I mean, it is impressive how well behaved the large genome serials are in terms of progression through meiosis, given the size of the chromosomes. But then there's other species with far larger chromosomes, and they still manage to progress normally. So it's probable that Arabidopsis isn't a great model, surprisingly enough, to really take apart how this works on a large genome species. So it's I think yes, you're probably right. There's chromatin and chromosome structures aspects that we probably don't really understand at all.
Luke, have you considered looking into natural variation for RECQL4, and maybe it's a highly conserved gene? Would this be an ideal candidate to look into structuration in regulatory sequences?
We have looked at an initial screen for some natural variants within both landrace and spontaneous material. We have seen some variation. It's unclear whether that would be have a strong functional impact, but then the studies in Arenosa, which showed effects of things like on ASY1 on recombination, you wouldn't have guessed that a priori from looking at the sequence variation. So it's worth looking at, and again, looking at structural variation ahead. It's, yeah, indeed.
Thank you, Laura, for the introduction and for this opportunity to present some of the work that we are doing in my group, where we are actually concentrating precisely on the stages of stamen and pollen development that start when meiosis is finished. And that is exactly at this point, stage Wellington stage 8. You can see those tetrads that are just a product of meiosis, and from there on, after they are released, they very quickly build a very thick wall that is required for survival. And from there on, they will undergo a series of processes until final, what we call Wellington stage 975, with anther opening, the pollen is released on the way to fertilization. So we are interested both on these different stages of anther and pollen development, which we call maturation, and the process of anther opening itself. But today, I want to tell you about our studies on the process of pollen maturation. And just to briefly introduce this, I have these beautiful transcription electron microscope images taken by our colleague Ulla in the Institute, where you can see three important aspects of pollen maturation, which include this vacuolization. So there is this vacuole that grows stage by stage, but then when the starch, which is visualized as these translucent structures, starts forming, then this vacuole starts shrinking until it's completely resolved as the starch accumulates, and in turn, the starch is completely filling the pollen at the stage 9.5 right before the anther opening. So when the pollen is ready, it is engorged with these starch granules, which are a hallmark of cereal pollen, and that is essential for pollen viability. The other process that, of course, is very important is the mitosis. And So the haploid pollen starts as a single cell, but during these maturation processes, undergoes one mitosis round to generate a vegetative cell and a generative cell, which in turn undergoes a second round of meiosis to produce the two sperm cells, which are the actual cells that are going to fertilize the female. But of course, the vegetative cell is the workhorse machinery that is keeping the pollen alive and that is doing all sorts of metabolic processes that are running the house, let's say. Now, how do we go about studying these processes in any organism? Well, of course, with our favorite subject, which is genetics. And for that, we've been very lucky to come with this excellent collection of meliceral mutants. And again, a big shout to Jerry for creating these great introduction lines in Bowman. And this includes this mutant MSG38, which shows a complete sterility. So I mean, maybe it's not so clear from these pictures, but this spike is completely empty in the mutant. And we quickly realized that even if the stamens seem to have a relatively normal morphology until very late in development, and this is basically stage 975 when we observe opening in the wild type, this would never occur in the mutant. So at first we thought, oh great, we have an anti-opening mutant. But actually, when we look more closely at the pollen, we quickly realized that pollen grains, although they look alive according to this Alexander staining, and this purple staining indicates that the pollen is somehow alive, but it looked quite strong. And moreover, when we use Lugol to stain the starch, we noticed that it was completely devoid of it. We followed this process of starch accumulation also in histological sections, and as I indicated before, this is already starting around stage 9 in the Bowman background, and by the stage 9.5, shortly before anter-opening, the pollen is filled with it. So it's this purple staining here. And this we are not able to observe in the mutant at all. The other thing that we notice in particular is that while the vacuole resorption was happening quite well in the Bowman, didn't seem to occur normally or at all in the mutant, where even until these late stages of development, you would still see those big vacuoles there. And we confirmed this data with transmission electron microscopy, where we could see beautiful plastids full with starch, and in the mutant, either we didn't observe any starch at all, or sometimes you will see a few plastids with some starch granules, but they will never fill the plastid completely. Now, we went on to check if the mutant was affected in mitosis, because this is another process important for pollen maturation, and it was a chance that it was affected on it. But actually, these Dapier stainings show us that both Bowman and MSG38 reach the tricellular stage, so both of them are producing the vegetative nuclei and the two sperm cells. So in principle, these defects on the MSG38 mutant seem to concern mainly the phase of starch accumulation in the pollen. Now, because the mutant is fully sterile, it's a pain because we need to propagate it and maintain it in heterozygous populations. So we've done a lot of genotyping the last 6 or 7 years that we work with it. So we have plenty of data to notice that actually, on top of being only foundable in segregating populations, it doesn't segregate mendelianly. So we don't find the traditional 25% segregation that you would expect for recessive mutations, but actually reduce 16%. And this suggested already that this is a gametophytic mutation. So there we went on to check the pollen of heterozygous plants. And you can see already in these pictures that while Bowman pollen looks quite uniform, in the heterozygous plants, we had quite some pollen grains that were shrunken or apparently smaller in size. And my postdoc, Pika, who worked on this project, actually went on to quantify that. And we could see indeed that both pollen have a large number of pollen grains that are, both populations, have pollen grains that are quite normal, but the heterozygous plants carry a population of small pollen grains that we are actually quantifying also here, and that was not, say, observable in the Bowman when we do the cutoff right here. And additionally, we see a large proportion of shrunken pollen grains in these heterozygous pollens. And this somehow confirmed this idea that the mutation is affecting the pollen, the gametophyte, although, of course, some of the pollen grains have to be, mutant pollen grains have to be viable, otherwise we will never be able to obtain homozygous mutant plants. Okay, so we went on to clone the mutation, And this was actually the fastest part of the whole project. Thanks to these introduction lines in Bowman, it was fairly easy to learn that the mutation is affecting this gene encoding a JOCA enzyme in the auxin biosynthesis pathway. So the original mutation is a 1 base pair deletion, and we went on to create 2 other mutations in the golden promise fast background, by the way, which is the one we are using for all our plant transformations. And they confirmed that this was the correct gene. And just to give you a very quick reminder, these JOCA enzymes are basically the second step and the final step, in fact, in the synthesis of bioactive auxin. So auxin is made from tryptophan. It's just transformed into IPYA by these TAA enzymes. JOCA transformed into the bioactive IAA, and normally this IAA is very quickly conjugated or oxidized to what is believed are non-active hormone molecules. So, well, I just said that this is involved in auxin biosynthesis, but of course we had to show if this is really the case, and we went on to collaborate with Karin Lune and perform some auxin measurements, and what we are seeing here in blue is the values for Bowman, and in red, the values for MHC38. And here we are following the Waddington scale, and you can see very nicely how from stage 8 on, the levels of auxin or bioactive auxin, IAA, are actually increasing exponentially in Bowman, and this is a logarithmic scale actually. And in the mutant though, this occurs very mildly. So there is actually, at the end of the day, an almost 10 times more or 100-fold difference in the levels of the hormone. And then when we look at the precursors of auxin, tryptophan and APOA, we would see the opposite trend, right? Actually, the mutant was overaccumulating, particularly at the late stages, the precursors like tryptophan and IPOA. So this somehow was confirming that indeed this enzyme is failing in this mutant, and therefore IAA is not overaccumulating. In addition, we noticed and were surprised to see that, of course, because IAA is not accumulating, then there is also not production of the final byproduct, which is the oxidized hormone. And while the wild type was accumulating tons of it, the mutant didn't have any, of course. But more strangely, this non-functional version of the hormone is actually accumulating at much higher levels than the functional one. So the hormone is very quickly being degraded into non-bioactive hormone, and this is quite well known in rice, for example. Too much auxin is also bad, so therefore there is this mechanism to get rid of it as soon as it's done its function. Importantly though, I just mentioned that the phase of starch accumulation is happening at stage 9, or starting at stage 9, and this is also where we have this, very, very rapid accumulation of auxin and reaching those very high levels. So we went on also to check the expression of the gene by qPCR, and we saw that this followed very nicely the trends of the accumulation of the hormone. So again, it starts at stage 8. Actually, sometimes it's not even detected, but when it starts being detected, then it increases also exponentially. And close to stage 9, it seems to be reaching the peak until it reaches the peak at 925. It goes down at 935, and then at 95, already when starch accumulation phase is over, the gene is not detectable anymore in Bowman, while it remains in the mutant, which is also, by the way, expressing the gene at much lower levels, and this is a normal effect of positive feedback expression of the hormone in its own synthesis, which is, of course, missing in the mutant. We also performed in situ hybridizations to see where the gene was expressed, and we were a little bit surprised to notice here in purple that the gene was not only found in pollen, where we expected it because this is a gametophytic defect, but we also found it in the vasculature and in some of the anter-wall layers like the endothecium. But when we performed, we created a transgenic line expressing the protein fused to citrine, fluorescence protein. We could see that, first of all, we could not find the protein before stage 9. So the first time we see this at stage 9, then it becomes stronger at 925, 935, and we can even see it at 95. Here we have to actually do it in extracted pollen because the confocal is not able to penetrate the thick anthers. But what we see is that we can find the signal only in the pollen grains. So this somehow confirms that the active protein is only found in pollen, and it suggests that this somehow is confirming the idea that this is a gametophytic working protein. And it also suggests that the pollen is making its own auxin. Right? It doesn't need all this stuff that you see in other parts of the plant, transport and so on. The pollen is somehow receiving a signal and tells it, okay, time to make auxin, and this signal will go on and presumably help on the accumulation of starch, as I will show you in a moment. So we went on and did transcriptomic analysis and showed that of course there were plenty of genes that they were differentially expressed, but we did hierarchical clustering on the 3,000 genes that were differentially expressed. There were of course genes that were very highly expressed in the Bowman and then very lowly expressed in the mutant and vice versa. Of course, we focus in these two clusters here that contain the genes that are down in the mutant. And the cluster 1 had genes that were most highly expressed at stage 925 versus 935, so they were going down at 935. Well, here was the opposite trend, but in any case, both were expressed at lower levels in the mutant. What caught our attention when we did top-go analysis of this cluster 1 here is that actually there were a lot of categories related to energy production genes. So basically, we're talking about everything from sugar degradation, glycolysis, pyruvate metabolism, TCA cycle, I mean, you know, biochemistry 101. And then we could also visualize this here with these nice plant-seq plots where everything in blue indicates that these different steps in the basic production of ATP, from taking glucose and converting it into pyruvate, via glycolysis, putting it into the TCA cycle, and even the function of ATP synthesis. All that stuff was down in the mutant. And in fact, in contrast though, starch biosynthesis genes, there were only a couple of things that were down, and actually there were even a couple of things that were up. For me, what this told us was that what was affected in the mutant was not the expression of starch biosynthesis genes themselves, but the expression of the stuff that is needed to make starch, which is not only carbon, but also ATP in heterotrophic tissues, like pollen grains or roots, or potato tuber roots, for example. The main limiting factor on the production of starch is ATP. It's not the carbon. I mean, indirectly the carbon, of course, too, because that's the source of the ATP. And in contrast, though, we will also notice that sugar transporters, for example, there was a portion of them that were also down-regulated in the mutant, but another half seemed to be actually up-regulated in the mutant. So there the trend was less clear. To me, everything was pointing out that the effects of the lack of auxin were actually more downstream of sugar transport. So we went on to confirm some of these genes. I don't want to go into the details, but what I find quite nicely is that this is again the expression pattern of the MSH38 gene. Some of these energy-regulated energy production genes like cytochrome C, exokinase, etc., they tend to follow the same patterns of expression, also coinciding with the peaks of hormone production and so on, reaching a peak at 925. And again, these differences might not seem like much to you, but this is a logarithmic scale, and these are up to twofold differences in gene expression precisely at the stage 925, which is when the peak of starch production is occurring. So we went on and collaborated with Ali Ferni in the Plant Physiology Institute in Golm, because we wanted to ask, okay, are these effects in gene expression really resulting in changes in the metabolism? So this was actually very cool because, well, we went there with not much expectations, but immediately, for example, the data on the sugar levels. And these are 5 independent replicates in Bowman and 5 in the mutant, already indicated that the mutant is basically overaccumulating sugars, sucrose, glucose, fructose. So as indicated by the transcriptomics, the problem is not getting the sugars into the anthers from the mother plant. The mutant is receiving the sugars, but it seems that it doesn't know what to do with them. It's just dying in a pool of sugar. And in contrast, and as expected from the transcriptome, the levels of pyruvate were extremely low in the mutant, and so at least 2 of the very important citric acid cycle acids like citrate and succinate. So this somehow really supported the transcriptomic data, and I don't have time to show you, but we also now have proteomics data that confirm that just like the genes are down in the mutant, also the proteins of all these energy production pathways are down. So to summarize what I just presented to you, these are again another Lugol stainings, but now on the whole anther level. So before stage 9, there is not much starch in these pollen grains, but there is somehow a transition where the starch starts accumulating until it reaches really almost peak levels of 935. And our hypothesis is that the sugar is accumulating between stages 8 and 9, more or less, and maybe even widely, I want to speculate that maybe the sugar is actually stored in that vacuole, and this resorption of the vacuole is coinciding with the use of sugar to make the starch. Somehow there is a signal, maybe the sugar levels, that activate the biosynthesis of auxin, and in turn, auxin will boost the expression of those energy metabolism genes, which will increase the levels of ATP, which are absolutely necessary for the biosynthesis of the starch. So of course, nothing is happening in the mutant, and then you have very little or no starch at all in those anthers. So the take-home message that I want you to take today is that timely auxin accumulation shifts pollen development to a high energy production state that allows the accumulation of starch in barley anthers. And we got published this in Current Biology a couple of months ago, and we are naturally trying to follow this up because I think this pollen is a great system to study auxin signaling. Now it's a very simplified system with only 3 cells and only one probably involved in the actual production of auxin. And then we identify now an auxin response factor that I think is the one that is doing this job in the barley pollen. Okay, so I think barley is also a great system to do fundamental discoveries in plant biology, as this work shows. But of course, there is also practical applications to it. And something that is very interesting about this mutant is that in contrast to other auxin biosynthesis mutants that you see, for example, in Brachypodium or in maize, the plants look almost completely normal. I mean, they are normal, in fact. And the only fact is that the inflorescences don't extrude here, they are completely sterile. So the plants seem to be really affected only in pollen development, which makes sense if this protein is working only there. And that's nice in general, although for us it was a pain because we always had to genotype to identify the mutants. But it's good because it's also related to this idea that this protein is actually part of a bigger clade of yucca proteins that is somehow related to this clade of yucca proteins that contains both serial and monocot and dicot yuccas. But MSC3A belongs to a specific clade of serial yuccas that I think have acquired this specialized function of working in barley pollen. And I think this offers big potential to try to specifically block the activity of this yucca, and only this one, which is working only in pollen, to control the male fertility of cereal plants. Of course, this is for great interest in hybrid seed production. So if there is any breeder in the audience that wants to give me 120,000 euros to test this idea, you are welcome to contact me. I just want to thank the people in the group, Dika, who was the main driver of this project, and my star in the lab, Edelgard, who was creating the transgenics for us, and everybody in the institute, my collaborators, and Maria, actually particularly because when my group started, they gave me a lot of nice support and bioinformatics, pipelines, and so on. And thank you for your attention.
Great, Bob, thanks. Have you looked on the cell wall invertase in these pollen grains? Because it kind of reminds me work was done on the Lin7 in tobacco and in tomato, and they showed that it leads to male sterility.
I mean, as I say, yeah, indeed, if you pay attention to that summary of the down genes, indeed, there is a bunch of what I call sugar degrading enzymes, and that includes cell wall invertases and vacuolar invertases. So we have now a pool of very cool candidates that probably are induced by auxin to also, so on the one hand, I think the sugar transport is not affected, in fact, but the degradation of the sugar is already the first line of problems that we're having, but we haven't really looked into that. We can now create CRISPR mutants into those things and try to really pinpoint it. My money is on a vacuolar invertase or related. That would be my first candidate, but that's just a wild guess.
I would suggest to talk to Ali Fernie. You collaborate with him already, and ask him about the cell wall invertase role in this kind of thing.
Yeah, I know that there is a couple. I mean, in cereals, for example, these cell wall invertases are super important for the starch accumulation in pollen under cold stress, for example. So that already points at them also as good candidates for this. Yes.
Thanks.
So if you water with auxin, can you reverse the sterility?
No, but so cannot either the Arabidopsis mutant people. I mean, there is also a fertility phenotype, although completely unrelated to ours, in the yucca2, yucca6, the cousins of our gene in Arabidopsis. Also there you have a fertility phenotype, and it doesn't work. My hypothesis, and I think their hypothesis too, is that you need a very particular vehicle to make sure that the auxin is able to penetrate through all the tissues and through the very thick pollen wall in order to be active. We tried to do it in vitro, so we tried to cultivate the anthers and see if we could feed auxin. It didn't work. I mean, we were doing that while the paper was in revision because we were always afraid that they would ask us, okay, can you actually show, I mean, see, all our evidence, if you think about it, is a little bit circumstantial, right? We don't have concrete evidence that auxin is activating the expression of these genes. So it would have been nice to be able to have an in vitro system where you apply auxin and then you see a regulation of the expression, but we don't have that yet.
There are many MSG mutants. Which is the next one?
Well, I don't have time to show this here, and unfortunately my people are not here with me, but we do have 3 open mutants. 2 of them, I guess I can tell you already because the paper should be submitted soon. 2 of them are actually encoding cell wall degrading enzymes that are important to soften the tissues to create a pore, and one of them is important for the formation of secondary thickenings in the endothelium layer, and that's actually a very exciting mutant. I think that has something to do also again with metabolism, so we are collaborating with Ali there. It seems that this protein is important to have normal photosynthesis in the endothelium, which is providing the carbon to make lignin for the endothelium layer.
Yeah, thanks for the nice talk. I would like to extend Matt's question. Did you also test synthetic variants of auxin? Because these are quite well known to systemically distribute very well within the plant.
We did, actually. We tried also actually, I had forgotten about that because this was some of the things. I mean, my postdoc spent the first 1 or 2 years actually not so rewarding time because she did hydroponics, for example, with these things. So we grew the plants in a hydroponic system, we feed the hormone to the roots, but they were doing nice things to the roots, but they were never reaching the shoots. And again, I mean, for sure, there is, the auxin is always trying to go down anyway. And as our work really clearly shows, the auxin is actually made, I think it is made in the pollen grains. So I think it is quite a big deal to try to get it there. And again, that also, I'm shooting myself in the foot because I say we can make inhibitors to block pollen fertility if we block this enzyme. But of course, you will need a very good vehicle that will allow any inhibitor or auxin to penetrate these tissues. But I think it should work. That's, I mean, my hypothesis of why heterozygous plants are able to produce viable pollen is because the auxin from the wild-type pollen is being exported and is being transported into some of the mutant pollen which manages to survive. So it should work, but we need to know what vehicle and which hormone to use, yes.
Hi, everybody. Thank you for the introduction and for the opportunity to present my work. First, with the availability of the full barley and wheat genomes, as well as a wide variety of various transcriptomic data sets, now we have the opportunity to study comparative transcriptomics and especially in closely related species to study their similarities and differences and thereby expand comparative genomics by means of a functional angle. So in my talk, I will introduce you to a systematic comparison of barley and wheat transcriptomes and how this can offer some insights into flower development. So barley and wheat are highly similar in their morphological and developmental features, but they also share a common ancestor 40 million years ago. And despite their similarities, they also have important differences, as we just heard about the terminate and indeterminate nature of the spikes. The recently established genomes and pangenomes makes their deeper transcriptomic investigation possible. Besides all of this, the ploidy difference could explain how gene duplications are retained during evolution and how functional diversification of homologous genes happened. In our transcriptomic analysis, we had to compile an RNA-seq dataset, for which we use the mining of public data. And for which we used the mining of public data, and we had 3 main objectives. And the first one, that I aimed for a large and representative data set that can mask minor differences between variety or technical differences. And so I collected in total 1,095 samples. And on this style representation, you can already see that inflorescence is quite abundant across the 2 species. But we have a minimum of 50 samples for each of the major tissue categories, grain, root, and shoot as well. My second criteria was that each of the samples had to pass this stringent selection, where they had to be non-treated and wild type, and they had to be part of an already peer-reviewed published data set, and they had to come from a uniform sequencing platform. And my third aim was to collect as many tissues as possible in order to differentiate between housekeeping and tissue-specific gene functions. So I in total collected 58 tissues, from which 13 tissues are present in both species. And on this figure, I grouped these 58 tissues to intermediate tissue categories. So you can already see there is quite a few overlap. But still, there is few tissues that are present in either of the two species. However, as in the symposium it was reported earlier, for one or the other barley transcriptomics data sets are soon to be available, and that will be an opportunity to close these gaps further. In the creation of this data set, I was facing a challenge, particularly in growth staging methods. And it's a feature of using public data. So my samples came from various growth staging methods. The good old ZOC scale and the more recently established BBCH scale, a fixed scale which concentrates on the Northwest European crop conditions, and the Weddington scale, which was already introduced, focusing on flower development. Now, having samples from various scales meant that I could not use age as a meta-information across all of my samples, and this would be very important, especially when we want to focus on development or flower development. So by carefully reading the definitions, these staging methods, I established a conversion table. And to give you an example, let's take this definition. Flag leaf visible, fully unrolled, and it can also be described as the green enter stage. Now, if we put this definition to the methods, it would be equivalent to 39 in both ZOC and BBCH scales, which will translate to 9 at fix, and in the Weddington scale, it would be in the range of 6.5 to 8.5. And in my approach, ZOC scale was the best because it was including the most variety of sample differences. And this table will be also soon published and available, but should be first consulted with, confirmed by consulting breeders or other experts. Now, after compiling this RNA-seq data set, we used several methods in order to be able to compare barley and wheat transcriptomes. And following the necessary pre-processing, we established a list of orthologs across the two species. And in the final steps, we conducted weighted gene co-expression network analysis, which was also introduced and mentioned in earlier talks. And it's a well-established method. And the final steps, we investigated module conservations across the two species. An important note in pre-processing that although I had several varieties for both species, I mapped all the samples to a single cultivar, Morex in barley and Chinese Spring in wheat. And after several steps of data curation, I'm happy to give you the integrity of my data set, where in the SPCA you can see in 4 colors the barley in orange and the 3 wheat subgenomes in other colors. And there are ellipsoids drawn around the centroid of each of the data sets. And as you can see, you cannot see much difference. And this is exactly what we hoped for. There is a big overlap between the two species, which means that the differences between samples are not defined by the species, but rather something else. And that is, as visible in this UMAP on the right, the tissue differences. So UMAP clusters similarities, and here the colors represent the 4 major tissue categories. And you can see some clusters are further away because, as I mentioned, it's a highly varied data set, and there is some overlap as well. It is not all microdisected RNA-seq data. Now I would like to walk you through the ortholog analysis. So besides the diploid barley, we have an allohexaploid red wheat genome with the A, B, and D subgenomes. Now for Morex, there are 36,000 genes, and for Chinese Spring, exactly 3 times more, 108,000 genes. We used a tool called OrthoFinder to define 25,000 orthologs across the two species, which is quite a big portion, around 70% of the barley genome and around 30% of the wheat genome. But within these orthologs, we had to subset the single copy orthologs, which are present in each of the chromosome pairs exactly once. And this gave us around 11,908 genes or orthologs. And while there are other combinations of orthologs present in the two species, in this analysis, I will only focus on the single copy orthologs. In the results, I would like to first give a global overview about barley and wheat networks, and then an example for each of the conserved and diverged modules. So on this circular representation, the four big parts represent each one of the orthologs. So the blue color, the orthologs from barley, and the shades of purple are the orthologs from the 3 wheat subgenomes. And within each of the bigger segments, the boxes represent the co-expressing modules of orthologous genes, which means that in this figure, each gene are represented 4 times. If you want to see how they are shared or how they are linked between the different subgenomes, then we can add different representation to these links. And on top of that, we could correlate modules with tissue. We could correlate module expressions with tissues. And here the outer circles are heat maps of p-values of module tissue correlations from outer root, shoot, brain, and inflorescence, where the darkest shade of a color represents that there is a strong correlation with that tissue in that module. Now let's take a closer look to the barley co-expression network. In total, we have 34 modules, and what is already clearly visible that each of the modules are strongly correlating with one of the tissue types. And then if we do additional validation with geo-enrichment, for example, here module 1, 3, 4, 6 are clearly correlating with flower, or here module 10 and 30 are clearly correlating with brain tissues. And then if we take a look to the wheat network, we see some similar patterns, but in total 23 modules. And again, we could do a geo-enrichment of each of the modules and see how they are correlated with tissues. And within wheat, the comparison is much more similar as it was expected. So for the B subgenome, we have similar number of modules, just as well as for the D subgenome. Now, while there are many interesting examples that we could take a look, let's just focus on one and how that would give more insights to this comparison. So here, the first and biggest modules of the A subgenomes, of the B subgenomes, are spanned across the barley network. And you can already see a comparably strong correlation with inflorescence with all of them. And if you take a different angle to the same example, then you can see that the first modules of the wheat subgenomes are not just overlapping with the first barley module, but several others, and the biggest overlap are in the first 3 barley modules. But barley module 2 is strongly correlated with shoot instead of inflorescence. And that part of overlap is exactly 156 genes that can be a potential target for further investigation. So in summary, I wanted to show you how systematic comparison is possible between two species to narrow down our focus to a subset of target genes. And this was possible mostly because of our large tissue selection. And we could see that the ortholog expression modules are comparable across the two species. They are strongly correlating with tissues. And these candidate gene groups can be investigated further in later analysis. And in the near future, I would like to include the developmental age because it would give even more insights. And I would like to have a more systematic approach with graph encoding to find the subnetworks of these overlapping modules. And I would like to include the other ratios as well. And in a far, far future would be great to extend this analysis to further 3D species. And with this, I would like to thank for the support, always encouraging support of my group, especially my closest supervisors, Georg Haberer, Klaus Meyer, Manuel Spanagol, and Nadia Kamal. And thank you for your attention.
Thanks a lot for a nice presentation. Great idea to use the resources and reanalyzing them, I think. I probably missed this, so can you say again how you defined a module, and even more so, how did you define whether a module overlapped or correlated in barley versus wheat? I mean, what were the criteria to say that the module is the same?
Yes, so when we did the ortholog analysis, we defined orthogroups across the 4 subgenomes. I just refer them as subgenomes. And then when I created these circular representations, then each of these segments are the orthogroups divided back, or let's say placed back to the original gene in barley and wheat. So it is between the modules there is no correlation. It is just, is that same ortholog present here or present there? If there is if they are present here and there is a connection between them.
I just have to ask again, so a module is not a set of genes that is co-regulated. That's not correct.
So just like in double VGCNE, as the method gives out co-expressing modules, yes, each modules are regulatory blocks. But then the members of modules across the 2 species, they are just in this representation, they are just shown which modules are they in the different species. So,
my question was, when is the module the same? I mean, how many genes need to be in this co-expression cluster, need to be overlapping between barley and wheat?
They are not the same. So, for example, here, what you see is that the wheat A, module 1, has this many orthologs present across so many other barley modules. So they spread across barley modules. Yes. Thank you.
Thank you. Thank you, Laura, for the introduction. I'm very pleased and honored to be here to present our latest results. So well-informed plant geneticists divide the breeding history basically in 4 phases. First phase, which is the one when we had domestication, and then the second phase of about 100 years when we did crossing and based on Mendelian genetics. And the third phase when we are now, and where we are doing crosses based on molecular genetics, using markers, using genomics, and using genetic engineering. And so we probably are going to be able to move too soon to the 4th phase, where we should be able to create the best combination of alleles for each of the most important genes that we think are important to improve our plant. So, considering trying to apply this vision to roots, we see a lot of constraint, a lot of limitation. We basically know very little about the most important genes, and actually we are still discussing about to define the phenotypes. When we are look and we think about the root types in cereals and in barley, we have to remember that we are talking about 3 different types of roots. The seminal roots, which are the roots coming out from the embryo, also divided in primary roots and seminal roots, which are the one coming from the scutellum zone of the embryo. Then we have the crown roots, which are the roots coming out from the nodes of the stem. And the third type of root, which are the lateral roots. We define lateral roots, each root which is coming out, developing out from a different root. Okay? So all of these roots, they put to, the structure, the length, the gravitropism, the branching of the roots define the root architecture of a plant, which you can see it's very vaguely defined. And this is basically when we enter into the difficult part. So when talking about root architecture, we are basically talking of nothing. So we have to subdivide and target our attention to the most important of these possible traits. And here I just put down a list of these possible traits, which is definitely not a final or a full comprehensive list. We are talking about the ratio between biomass between the root and the shoot. We have to deal with the anchorage function of the root and the root lodging. We have to think about the root spatial architecture, as I said, which is very easily subdivided in many different components. I don't want to enter into the discussion now, but you can see that, and we will talk about a lot later on about gravitropism. And then we have all the anatomical traits, and we have been also hearing yesterday, if I remember well, about the parenchyma, and about the xylem vessels and so on and so forth. We have to deal with root hairs, with root exudation and the carbon allocation derived by the root exudation, by the soil exudation in the soil. We have to deal with the rhizospheric microbiome, which is actually very much related with root morphology and function. And of course, at the top of it, all the function related with nutrient and water absorption. So just to underline, I won't make here any list of genes or QTLs which have been mapped in the last few years in roots, but just I wanted to highlight it that if you go to the Barley Genetic newsletter and you search for root and root genetics and root mutation, you only find one single mutant. And this mutant is called fewer root 1, and it's basically quite an interesting mutation. You basically lose all the type of mutation, type of root that I told you before. So the root coming from the nodes, the crown roots, you only have the seminal roots. And unfortunately, the seed stock of this only single mutant was lost. It was lost a few years ago, and Uda told me a few years ago about that. So we don't have any, formally, we don't have any root mutant in the seed stock. We have to be more precise, we have root stock of mutation about root hairs, but not about root architecture. So in Bologna, we developed already many years ago a mutagenized population, and chemically mutagenized with sodium azide. The population was originally 5,000 lines, and now after a couple of cycles, 3 cycles of multiplication, we only have 3,600, more or less. We use it for tilling, but during the many years of working with this population, we realized that there were so many phenotypes. When we had the plants in the field and we are looking for phenotypes in the shoot, we realized that we had so many, basically more than 1/3 probably of the plot had one phenotype, one clear phenotype, that we decided that the population should have also be exploited in forward genetics rather than in reverse genetics. And you can see an example of this exploitation in poster number 72 by Serena, who is here. We screened the population for root phenotypes. We did it at the beginning with very simple approach, like the very famous paper roll or paper cigars. Then for the most important phenotypes, we did a second screening using a flat perspex type of rhizotrons. And for the selected one, we also use soil-filled rhizotrons. we ended up in having a collection currently of 34 root-specific mutants, which we broadly classified in 4 classes. Hypergravitropic mutations, and we have 3 of them. Short root mutants, 23. Short, like disturbed, wavy, and with 4. Of course, the boundary between this one and this one is very debatable. And also we have now at least 4 mutations for root hairs, both short root hairs and root hairless. We definitely decided to try to clone at least the most important one, and currently we clone 4 of them. So I'm reporting the latest news about the cloning of 2 of them, which are the hypergravitropic one. So the first one was, we finished the work less than 1 year ago, last year, and we published on PNAS and the cloning of the gene that we called enhanced gravitropism 2. Here you see a phenotype, a typical phenotype in blue paper after 5 days after germination. So here you see Morex wild type, and here you see the mutation. Here you see a phenotype in after 3 or 4 days after germination in using MRI, magnetic resonance imaging. And so the gravitropism was fully confirmed. We fully confirmed also the gravitropism using a typical rotation test that you use to test whether you have an active reaction to gravitropism and not a passive one. The gene was mapped and cloned using a very standard bulk segregant analysis using high-density genotyping on an F2 population. The population was created by crossing the mutant lines with the bark in order to generate the polymorphism. Then we also whole genome shotgun sequenced the mutant line, and we found, based on the BSA, the mutation was placed on the chromosome 5. And by sequencing the line, we discovered 6 mutations very close to the peak, but only one was carrying a stop codon. So we had only one line with only one phenotype, so we couldn't really be sure that that was the gene. In order to get at least a second allele, we used gene editing, and so we generated the second allele, EGT2/2. And in this case, we targeted the first exon. And this is the phenotype of the second allele, again, hypergravitropic. The gene is there. The gene is controlling root angle in all the root types. So seminal, crown, and also lateral roots. Here you see a close-up of the lateral roots, which are also hypergravitropic. The gene is expressed in the whole root tip. So you, for people not in the field, you divided the root tip in the columella, in the meristematic zone, and in the extension zone, where you get the elongation of the cell. Well, the gene is expressed basically in the second, in the meristematic zone, and in the elongation zone. And most kind of broadly expressed in all type of cells. We also did RNA-seq analysis, and with these 3 type of tissues from the root tip. And we realized that comparing Morex and the mutation, we found 67 differential expressed genes. And with our surprise, most of the gene differential expressed were related with plant cell functions, like expansins and also peroxidases. It was interesting to note that this gene has a close homologue, which we are not really sure whether it is the ortholog or not, because the phylogenetic distance is very large. But it was known that a similar gene, a sterile alpha motif gene, which is the main domain of this protein, was also found in this gene controlling shoot gravitropism in pitch. Then, if you remember, I said at the beginning that we had 3 different root hypergravitropic mutations, so we also tried to clone the other 2. And this ended up in a second nice work, which was just accepted very, very recently, so I'm not sure whether I'm allowed to tell you, probably, but anyhow, I go ahead. And so you see, that, again, the phenotype is pretty much similar to the first one. We call this second gene EGT1. And here you see a phenotype in a a rhizotron, in soil filled rhizotron, where the different type of roots were colored with different colors. And again, you can see that the gravitropism is altered both in the seminal roots, the crown roots, and also in red, the lateral roots. The gene was cloned using the same approach. We ended up, in this case, we had two lines, so two alleles from the very beginning. And the two alleles, the two lines were sharing, they had hundreds of mutations, but they only shared one single mutation in one single gene at the position, at the locus, at the target locus, which was on chromosome 6 in this case. In this case, the gene encodes enough box TAPI domains, which is known to be basically a many-function protein. So some of them are transcription factors, some other are involved in protein-protein interaction. In this case, differently from EGT2, we searched for a natural variation for this gene, and we found, when we used the Welby collection in collaboration with Luigi Cattivelli from CREA, and we were able to see a nice presence of 6 prevalent haplotypes and a comparison between haplotype 2 and haplotype 4 in the Welby population showed a statistical difference in root angle. So this is probably a gene which is involved also in controlling and adapting root architecture in the environment and possibly in the cropping system. EGT1 acts completely independently from auxin. We ran all the experiment with treatment with auxin and with NPA. I don't want to waste but this was very clear. We also ran all the gene expression analysis using RNA hybridization, and it was very clear that in this case, differently from EGT2, the EGT1 gene was expressed in the vasculatory region of the root, especially in the elongation zone. The RNA-seq told us again that, like EGT2, the type of protein and genes which are differentially expressed between the mutant and the MOREX were mainly function related with cell walls. This started to generate an hypothesis. Probably these two genes, EGT1 and EGT2, are involved in cell wall flexibility, which are basically the counterpart of the gravitropic force generated by auxin. So to test this hypothesis, we decided to test whether cell wall of the mutant were differently in strength, in solidity compared to the wild type. So we use an atomic force spectroscopy method, and we revealed that indeed the mutation EGT1 produce a less stiff cell wall in the cells of the tip of the roots compared to MOREX. I can skip this. So going back to, so let's step back 1 minute and see how gravitropism works. So the gravitropic force is perceived by the columella cells with the statolites, which are moving down in the direction of the gravitropic vector, and that movement generates a signals in the cell, which translates basically in a strong auxin flux going down in the lower part of the roots, and that until to the cell elongation zone. The high concentration of auxin in this region will reduce cell elongation. The opposite phenomena will happen on the opposite side of the root. The minor concentration, the lower concentration of auxin will generate a longer and stronger cell elongation. That is the mechanism of bending of the root in respect of the gravitropic force. So this is basically an explanation that biologists used to explain gravitropism, but it's very clearly a very limited explanation, because if this was the only force acting, all the roots would be perfectly vertical. Okay? But this is not the case, because every root will grow in reality using following a gravitropic set point angle, where definitely you have the gravitropic force, but you must have also the anti-gravitropic offset mechanism, keeping the roots at a very defined root growth angle. So this is basically what we found by finding and cloning the two genes, the two EGT1, the two EGT genes, because we are basically, those two mutations are acting on, affecting this anti-gravitropic mechanism, which is basically not functioning into the mutation, leading to a very vertical growth of the root. So the two genes are basically, so they're doing more or less the same at the level of cell wall. EGT2 seems to also to have, based on RNA-seq data, some feedback to the PIN, so to the auxin mechanism, but the two genes, as I said, work together in this anti-gravitropic response, which is basically the first time that a gene was, or two genes were clearly cloned to be acting on this regulatory mechanism. We carried on and tried to see whether these genes function also in wheat, taking the advantage of the availability of the Tilling population in GIC from Cristobal Huawi, and indeed, after some effort of the group, they were able to provide us with the triple mutants in cadenza for the EGT1 and also for EGT2. And with very much happiness of everybody, the phenotype was there. So the genes can be used to change the gravitropism also in root of the wheat, which is one of the most important crops. And so perspective, definitely now we're trying to go to the field. All this was done in a rush in basically control condition. We don't know really whether these genes and these alleles, what they do in the field. So we are already at the backcross 2 conversion using elite Italian barley lines for EGT1 and EGT2. And at the same time, JIC and NIAB in Cambridge are doing the same for durum and bread. Of course, having cloned the two genes, we should open the door to understanding better the second mechanism controlling gravitropism. We are doing that also. We are not checking root gravitropism only in the mutant population, but we are also using genome-wide association. Please go to see poster number 51 by Giuseppe Sangiorgi, who is here. So I don't just want to lose any time. Just wanted to tell you that we are moving to screen and starting to screen the Tilmore population again, with more fancy, possibly high throughput methods. We are using these CD cases in order to be able to screen 1,000 plants per day or per week, let's say. And we are looking for the opposite type of phenotype at the moment, so the ones that are not actually sensitive to gravitropism, so the agravitropic mutants, in order to find new genes. So I'd like to finish to thank my direct collaborator, Giuseppe and Serena, who are here, who are the main responsible of this work, and a very nice collaboration with the University of Nottingham for EGT1, University of Bonn with Franco Koldinger for EGT2, and all the other colleagues who provided help or support. Before I finish, I was asked to inform you that we are organizing a very nice conference at the end of this year in October 2022, which is going to be dedicated to geneticus, durum, wheat, and pasta. Thank you very much.
Do you see any signs that there are homologs elsewhere in the plant kingdom with the same function as EGT1?
Say it again.
Elsewhere in the plant kingdom that have the EGT1 function that are homologs?
Yeah, good question. The two proteins, the two genes are very well conserved. Yeah. Very well conserved until you get to the algae. So there seems to be something, if I read in your mind, connected with the movement of the plants out of the water. Yeah, you're right.
Just a quick question. Did you ever see something like FAIR1 again when you screened your Tilmore collection?
FAIR?
The original mutant, did you see that again, the FAIR1 that was lost from the collection?
That's a good point. No, because we are not, at the moment, we are not growing the plants to that point. In order to get the high throughput, and we are keeping the plants only for 1 week or more, so you don't see that, because you have to wait until you get the crown roots. Yeah, but that phenotype is actually very similar to a very famous mutation in maize, which is called RTCS, which is basically exactly the same phenotype in maize. So I can bet it's the same gene. So maybe we can prove it by gene editing. Thank you.
I just have a question. I missed it quickly when you showed the GWAS that you're planning. You did it on the Tilmore. And I thought you also showed that you're planning to do it on the Welby. And the question I had on this is, are you using rhizotrons for the phenotyping to grow them or just on plate? And what kind of root phenotypes are you scoring for in your GWAS?
In the GWAS, yeah. We have Giuseppe and can be much more informed than me, but we are actually using this sort of midway type of rhizotron, which are 40 centimeter times 30 centimeter rhizotrons, and the plants are kept for 7 days. And so you basically see all the seminal roots until they start to develop the very first lateral roots, but you don't see the crown roots, as Alan was asking.